So far, the DNA molecule has unwound, the hydrogen bonds between the bases have broken, and now the bases are exposed, just like in this diagram. As it says, this process occurs at several locations on the DNA molecule, and DNA polymerase can only add DNA nucleotides in one direction. Now, this is really, really important. This results in one strand being replicated continuously and the other strand being replicated in fragments. Now, the one that's continuous is called the leading strand, and the one that's made in fragments is called the lagging strand. And we're going to look at the formation of both of these. So this rule is really important, that DNA nucleotides can only be added in one direction. So the first one we're going to look at is the formation of the leading strand. This one's a little bit more straightforward. So once the DNA has been unwound, the hydrogen bonds have broken, and now the DNA bases are exposed. So in the original DNA molecule we're looking at, this is represented by the blue bases. So the blue ones here are the original molecule, okay? So the first thing that happens now is a primer attaches. So a primer is basically a short series of nucleotides. It's just a few nucleotides, and the prime, if you think about primary, you went to primary before you came to secondary. A primer is the thing that starts the process. You need a primer for DNA replication to occur. As it says, a primer attaches or joins to the 3' end of the original strand. So we know that nucleotides can only be added in one direction. So if you have a look here, the 3' end on the original DNA molecule is here, and this is the 5' end. So the nucleotides can only be added to the 3' end first. So it can only go in this direction. Can't go in this direction, has to go in this direction because it can only be added to the 3' end. As it says, DNA polymerase adds 3 nucleotides to the new strand. So you've got your original strand and you're making up this new strand. So what DNA polymerase does, the enzyme, it collects nucleotides from elsewhere, 3 nucleotides, and it brings them here, and it basically pairs them up with their correct base pairs. And it does it, the way I'm using the mouse here is how it does it. Okay, it has to go in, starting at the 3' end, and it goes in that direction. Now, a good way to think about this is if you can imagine at PE when you have to do your Scottish country dancing, and no one wants to partner up, they might say, let's say they line up all the girls, and then the PE teacher goes and collects the boys and pairs them up. That is what's happening here, okay? So DNA polymerase is like your annoying PE teacher, and the nucleotides are like the reluctant boys that are getting dragged here and paired up with a girl, okay? That is what it's like. So, as it says here, synthesis occurs in a 5' end to 3' end direction on the new strand. So this is something people often get confused by. They're not sure which direction things are going in. On the original strand, which is the blue one, it's going from a 3' end to a 5' end direction because the nucleotides are being added to the 3' end first. The new strand, which is the red one here, it's going from a 5' end to a 3' end direction. That is where it's been made. So try not to get confused between that. Okay, learn the directions. Here we've got the DNA nucleotides will then bind to their complementary bases on the template strand. So once they're in place, they then bind to their complementary bases, and a hydrogen bond would then form between each of these.